these genes that are activated, these genes that are transcribed immediately after touching the Arabidopsis plant, Bram and her colleagues called touch genes. And the first of these touch genes that she studied encodes a protein called calmodulin. Now I know this is getting a little bit specific, but just bear with me and you'll see why this story is important for understanding plant responses to tactile signaling and its connection to human responses to tactile signaling. Calmodulin is a protein which is involved in calcium signaling within the cell. As I mentioned in the beginning of this lecture, calcium is one of the important salt ions that regulates a cell's electrical charge and communication between the cells. Calcium is the ion that's released when the action potential reaches the end of a nerve cell, causing the release of the neurotransmitters, enabling the propagation of the action potential to the neighboring nerve cell. So one of the first genes that's activated once a plant is touched is a calcium binding protein called calmodulin. In plant cells, calcium helps maintain the cell turgor, the pressure within the cell, like in the pulvinus of the mimosa. And it's also part of the cell wall. It actually has many, many, many roles. Although we don't know how all of these roles are integrated, it's under intense study in many laboratories around the world. We also know that following a mechanical stimulation, like shaking of a branch or when a root hits a rock, the concentration of the calcium ions peaks. It spikes very quickly and then drops. So this spike in the calcium concentration affects the charge across the cell membrane, but not only does it affect the electricity of the cell, the charge, it also directly affects multiple cellular functions, and it does this through binding of calmodulin. So calmodulin is a very small but important protein. And when it binds calcium, it affects many, many, many cellular processes. For example, in human beings, the binding of calcium to calmodulin is important for memory, inflammation, muscle function, and nerve growth. So getting back to plants, the first touch gene identified by Bram was calmodulin. In other words, when you touch a plant, be it Arabidopsis or papaya, one of the first things the plant does is it makes calmodulin. Possibly, a plant makes more calmodulin to work with the increased calcium that comes with the action potentials. So thanks to the continuing work of Bram and other scientists, we now know that over 2% of the 25,000 Arabidopsis genes are activated when the plant is touched. These include calmodulin and other calcium-related genes, but also a plethora of other genes. Think about it. 2% of the genes are activated when you touch a plant, when an animal brushes up against it, or when the wind shakes its leaves. This is a surprisingly large number of genes, and it just indicates how far-reaching a plant's response to tactile sensation has to be in order to survive. That the plant, rooted in the soil, has to respond in multiple ways in order to modulate its growth, in order to modulate its own physiology, in order to survive in a changing environment. So we've seen vastly different reactions to touch. We've seen fast reactions, such as in a Venus flytrap. There are slow reactions, which we haven't time to show, such as root navigation. There are very, very, very slow responses, such as thigmomorphogenesis. But what they all have in common is some type of electrical signal, and at the far end, some type of change in gene transcription.